Automatic data acquisition software is extremely necessary for collecting huge amount of cryo data for structural characterization of biological macromolecules.
Automated data acquisition software package was used here to characterize the structure of various biological macromolecules which are directly associated with pathogen biology, for example Mycobacterium, HIV and SARS-CoV-2.
To begin with, start Latitude-S automated data acquisition software by clicking on Digital Micrograph from the Start menu. Next, select the Technique Manager and then the Latitude-S icon for single-particle automated data collection. To create a session based on the previous session settings, check the Based on Prior Session checkbox in the palette and then select the New button. Or to continue an existing session, press the Continue button. To start an entirely new session, click on the New tab in the palette. Choose the folder containing the session to be continued and select a folder to save the data. Next, click on the Setting icon and in the Manage State Explore box that appears, add state, set the TEM condition, camera condition, and image or stack option, and then name the state or open the state setup summary to open the saved settings. Next, configure the Atlas state by clicking on